session and symposium 16. We have four speakers, and the time allocated for each speaker is 20 minutes. And I'm Professor Ranju Bansal from UIPS Punjab University. And sir is
Professor Anil Gaikwad from BITS Milan.
so. Okay. So we have with us first speaker, Dr. Olga Tarasova, and we welcome you, ma'am. Dr. Olga, it's my pleasure to introduce Dr. Olga, and uh, she graduated from Pirogov Russian State Medical University in 2008 and earned her PhD in mathematical biology and bioinformatics from the Institute of Biomedical Chemistry, Moscow in 2012. She currently serves as head of the laboratory for big data analysis in digital pharmacology at IPMC, established in 2024 under the National Program for Basic Scientific Research. Her laboratory focuses on big data analysis in medical virology to identify safe and effective antiviral compounds and optimize infectious disease therapies. Dr. Tarasova has authored and co-authored over 70 scientific papers and several book chapters. So a big round of applause for her. And she has 48 publications indexed in Scopus. She has led major research projects funded by Russian Foundation for Basic Research and the Russian Science Foundation, focusing on drug resistance and HIV host interactions. Her team developed the HIV host web platform and a computational platform for antiviral compound discovery. In 2024, she received the first Prigry Prize for Best Investigation at the Young Scientist Forum of the Russian Academy of Sciences for her innovative work on HIV host interaction analysis. So, mainly, she works on bioinformatics, cheminformatics, virology, antiviral agents, and big data analytics. So, we welcome you, Dr. Tarasova. So, you may start with your presentation.
Thank you very much. First of all, I would like to thank all the organizers for the opportunity to present our study, and it is a great honor for me to be on this conference, IPSCON. It's a very honored conference, as far as I understand. So let me talk about the prediction of adverse reactions of paired drug combinations and drugs based on structure-activity relationships and network approaches. So it is well known that the drug administration is associated with the risk, sometimes with a high risk of adverse drug reactions, and some of them may lead to the serious life-threatening consequence. And it is well known that use of multiple medications, sometimes polypharmacology, may induce drug-drug interactions, which may increase the possibility of adverse drug reaction occurrence and may increase their severity. And of course, it is important to analyze all these drug-drug interactions and to predict the possibility, the probability of such occurrence of such drug-drug interactions and adverse drug reactions. So we are now working on the computational approaches for predicting activity of chemical compounds, activity of drugs, and this work is focused on the development of computational approach for predicting adverse drug reactions for drugs and drug pairs. A few words about machine learning and artificial intelligence. They have the high role in the drug discovery. More and more, they became more and more powerful for drug discovery, for computational drug discovery. And in this review in drug discovery today, the authors analyzed various drugs, mainly biotechnology, produced by biotechnology. And they found that artificial intelligence method can help to increase the probability to discover chemical compounds that became drug on the latest phase of clinical trials. This is especially important because the cost of fault of wrong wrongly found the drugs, the chemical compound that can be active but can fail on the last drug clinical trial phase is especially important. So that's why it is important to find safe and safe drugs and drugs that can have the lowest probability to have adverse drug reactions. So let me start from our approach to predict adverse reactions of single drugs based on structure-activity relationships. There are a lot of sources that describe drug reactions. Some of them are arranged in the databases of adverse drug reactions, and the main the most important that these sources should in the adverse drug reactions in these sources should be standardized using MedDRA dictionary. And we use two database, the CIDR database and on-site database, which will be used in this presentation, this study. That contains adverse drug reactions described in the box warrant and warrant precaution sections. And they are standardized with a MedDRA dictionary, and some of them, most of them, are manually cured. To predict, we use this database of the source of the adverse drug reactions, and negative examples are those drugs which do not have adverse drug reactions in their list. So we use this set of positive examples, the drug with the known adverse drug reactions, and negative samples, the drug without these known adverse drug reactions. And then we use well-known machine learning procedure described, implemented in past program that is based on the Bayesian classification, and that presented the structure of chemical compound in the set of substructure descriptors. So then we can build statistics, and then we can predict the probability of the presence of a particular adverse drug reactions and the probability of its absence. So we used this program, we trained it using the set of drugs from the on-site database as positive and negative examples. And we use two threshold with P minus PI higher than 0.5 and higher than 0.3 to select the drugs that can have particular adverse drug reactions. And there is a accuracy receiver operating characteristics under curve that is about for the mean accuracy is over 0.8 for well-known and for rare adverse drug reactions. So there are some of them that we may predict using our computational approach based on past. In our computational platform that is developed for prediction antiviral chemical compounds, we also have a feature to predict adverse drug reactions. This is an example of prediction such reactions for rilpivirine. It's the reverse transcriptase inhibitor, which also used in combinations with other drugs, but it is characterized by the depression of CNS sometimes, and it may cause depression and sometimes even suicidal ideation. So we use it as example in our some of our applications to understand how it may be caused. Also, we have an approach to predict adverse drug reactions for drug pairs because it is important due to the indications of several drugs for several diseases. So to predict the adverse drug reactions that is caused by drug pairs, we used drug bank for positive examples of drug reactions for two drugs, and two size database that was used as a source of negative examples for drug reactions, so that we know that these two drugs, these drug pairs, do not cause the particular adverse drug reactions that were included in the positive in the data set of positive pairs. So we formed the training set, and then we use the combinational description that give us the opportunity to describe the pair of drugs based on the combinations, all the combinations between MNA for one drug and for another drug. So we can have all these combinations of MNA and use the same Bayesian approach to predict the probability to have an adverse reactions for the drug pair. So we use these values of accuracy and sensitivity specificity. It's important to know that when we validate our approach, we excluded both drugs, both of the pair from the training set to predict their possibility to have an adverse drug reactions, and then compare to the uh, mm, well-known reactions for them to calculate the accuracy. We also analyzed how we can use our approach to understand the most common adverse drug reactions that can be predicted, and new drug reactions that can be predicted for pairs in our analysis. So this is a heat map that rows of this heat map contains the various combinations, and the columns describe the various adverse drug reactions with the PAPI values, so computationally predicted probability to have the particular adverse drug reactions. So we found that for most combinations of antiviral drugs with antineoplastic drugs, we don't have any adverse drug reactions. Probably QT interval prolongation and also rhabdomyolysis will be and sometimes hemorrhage will be in the top of the predicted adverse drug reactions for such various combinations. There is the heat map with the particular combinations of um, antiviral, anti-HIV drugs, and various types of drugs with the minus PI value in the particular cell. So we also may have an view how they can be combined and which particular adverse drug reactions they may have. Um, Also, when we find particular drug reactions for a drug and drug pair, we would like to analyze the causes of these drug reactions, the potential targets that may be associated with this reaction. What we do in our computational approach to find such mechanisms, such molecular mechanisms, we analyze the results of prediction for a particular drug. This is an example for real PV uh, for particular molecular mechanisms predicted by PAS or that are not associated initially with adverse drug reactions. So this is a simple list of any targets that are predicted for which this compound is potentially active. Then we use two computational approach to find the drug targets, the genes that are associated with the so-called seed genes for the studied diseases that were associated from the particular adverse drug reactions. For instance, for CNS depression, for depression, we use the particular disease from this genetic database, such as depression. So we compared each adverse drug reactions with a particular pathological condition, such as disease, for instance, from this genet, to find the particular networks and genes that are associated with the known genes for these pathological conditions for this disease. And we used the network that were published previously to find these subnetworks and find important targets, important genes associated with these seed genes and potential involved in the adverse drug reactions. We used two algorithms, random walking with restart and diamond algorithm. First one can help us to find the potential targets that are associated, that are connected with the seed genes, but are not directly interacted with them. And the second one can help find the so-called functional models that are connected with the seed genes and can be involved in the particular pathological condition. So re- uh, when we obtained the results of our computational procedure for rilpivirine, then we ranged the targets during as a result for prediction by pass PA minus PI, then the prediction of the random walking with restart algorithm score, and then it was ranked using these two algorithms. And also we took into account the involvement of a particular target in the model according to the diamond algorithm. So we may range these targets, and then we may use them to analyze a particular network, signal network, to make a signal network analysis. So then we used pathway enrichment analysis. So based on these targets selected for such type of analysis for this particular study, and we found several pathways that some of them include more than 10 genes associated with these adverse drug reaction with this pathological process. And then we analyzed them um, and found that there is phosphoinositol 3 kinase pathway. We found that some of these targets are known to be rilpivirine targets. Some of them are are potential targets of rilpivirine, and some of these targets in this pathway is the known targets for regulation depressive effect, depressive conditions. Another one pathway also we found several targets that are associated with the action of rilpivirine, and some of targets are associated with depression. So using this computational approach, we can understand whether this drug can be involved in the particular adverse drug reactions, and what are the mechanisms of these kind of drug reactions. Then when we found these potential targets, we also used the network approach to find potential master regulators. This is a genes genes and associated proteins that can affect the transcription of particular genes, and then can be on the top of signal network regulation. So in that manner. So we used network approaches and the transcription data from gene expression omnibus and upstream analysis, computational analysis to identify such master regulators. And we found several master regulators that were also included in our network analysis and were included in the signal pathways associated with the depression. And then we produced several more computational studies to find the potential mechanisms of rhabdomyolysis, since it was predicted at the um, highest with the highest probability to be induced by rilpivirine with some other antineoplastic agents. It is important because sometimes antiviral compounds can be used, in particular, rilpivirine can be used as a part of.
Dr. Olga, uh, since of the time constraint, you have completed 20 minutes. Can you please summarize?
Okay,
Thank you.
just a minute. So we produced the same work and found some targets that can be associated with the rhabdomyolysis for this combination. The same. So to summarize, we have developed a computational approach for predicting adverse drug reactions for individual pairs, for individual drugs and drug pairs. And we found that structure activity relationships, along with machine learning and artificial intelligence, can be successfully used for making such predictions. And network analysis coupled with the machine learning procedure can help us to identify potential targets for the adverse drug reactions and the drug reactions of uh, combinations. I would like to thank all our lab for taking part in this study, especially Dr. Sergey Ivanov, who has the main role in this study. And thank you for your attention. Mhm.
Thank you, Dr. Olga. It was a nice presentation. So, anyone having any questions from the audience? So, we have 2 minutes. Quickly, we can ask 2, 3 questions. Any queries from the students?
Of the time, you have used computational model and machine learning to predict drug-drug interaction and adverse drug reaction. So something are known, there are adverse drug reaction, interaction checkers and all. How that is superior to this machine learning process? Can it predict in advance based upon the genetic makeup of the host and the drug taken and the possibility of interaction with other drug?
Mhm.
There is a genetic makeup of the host. You prescribed a drug that has different mechanisms and different mechanisms of toxicity. That is first reaction. There is a possibility of adverse drug reaction or side effect reaction. If he takes another drug, okay, now two drugs, same host, what will be the probability of predicting definite association of this adverse drug reaction?
Mhm. If
You
I
have
just
you have presented to some extent, but I want to
Mhm.
hear something.
Yes, thank you for the question. If I understand correctly, you are asking whether we can predict the new adverse drug reactions for drug pairs, right? Yes, we are focusing on such a purpose. So we want to predict new adverse drug reactions for drugs and drug combinations. To predict new adverse drug reactions from the drug structure, we use the only one structure of one drug. And to predict the drug adverse drug reactions for combinations, we use structure of two drugs and the same method for generating descriptor for MNA for one drug and then MNA for the second drug. And then we made all possible combinations. For instance, MNA1 for first drug, the first one MNA for two drug. So we concatenate all the descriptions, all combinations. So the first row with this first row, the first with the second, etc. Well, we have all the combinations of MNA for both drugs. So we have like a description of the total structure made from two composite drugs, right? So then we used the same algorithm as was here predict described. So we obtained the probability, the frequency of have adverse drug reactions for drug if it include particular MNA descriptors, and also we obtain the probability to have such an adverse drug reactions in general. So it is the number of chemical compounds that have adverse drug reactions and they do not have the particular adverse drug reaction. So it's a probability in general. Then we use the statistical approach to also we understand how many chemical compounds include the particular MNA descriptor. So we have the three probability, this is three frequencies, and then we made up the statistics and we have a kind of curve that describe the probability for a new compound with the similar characteristics, with the similar frequencies to have the particular adverse drug reactions. And if we have two compounds, then we have all possible MNA combinations, the same statistical procedure, the same curve, and the same, yes. Mm. Yes. For person, yes.
Yes, sir.
Yes.
We are running short. Okay.
Communicate. Yes. Yes, it's large scale prediction. It doesn't include the personalization at this stage. Yeah, you're right.
So I have a very small question, Dr. Olga. It was a very nice presentation, and you have predicted the adverse effects for the combinations, right? So I was just thinking, so why did you choose the combination of antivirals with antineoplastics? Is there any specific reason for that? And my second question for the students is that which software you used for the all the predictions?
Mhm. Thank you very much. Why we analyzed antiviral and antineoplastics? First of all, it's a good example because sometimes antineoplastics can inhibit the same targets. For instance, it is known that some antineoplastics that inhibit some kinases can be also active as antivirals. So there is a chance to find some antivirals from antineoplastics. And then because sometimes antineoplastics is used in combinations to prevent some viral association cancers, such as, for instance, Kaposi cancer or some kind of that. So that was two reasons. There were two reasons. And another question we used PAS that is freely available on waytodrug.com. And our, how to say, daughter daughter platform viruses is included in waytodrug.com. So it is also to use it for prediction versus drug reactions for antiviral compounds. But this is the whole data set, so you can use not only antivirals here, you can use any compound because it's specific.
Okay, thank you so
Thank you.
much.